So welcome everyone to the University of Washington Data Science Seminar. If this is your first time attending, we'll be using the Q&A feature, which is the double chat icon on the bottom right-hand side of your Zoom screen, where you see the panelists today. And you can type in your questions there, and then we'll answer them after each speaker goes. The other thing, before I forget that I want to mention is that I think there's something going on next Tuesday. I'm not sure. But we won't be holding a seminar next Tuesday, so that everybody has an excuse to go out and make sure their ballots are deposited. Okay, so with that, let's get started. I'm really excited today because we're showcasing two rising stars who are pursuing a PhD in the chemical engineering department at the University of Washington. Both of our speakers today, Hawley and Orion, are using data science in chemical engineering. But what you'll see is that they're using two very different components of data science to break down the barriers of discovery in two very different areas of science and engineering. So our first speaker today, Hawley, will be using image analytics to help design better pharmaceuticals and treatments for health outcomes. And our second speaker, Orion, will discuss his work using natural language processing to design healthier and safer chemicals. I'm particularly fond of this pairing today because it not only shows off the breadth of diversity of data science, so image analytics and natural language processing, but also what the breadth and diversity of chemical engineers do. Okay, so without any more talking from me, other than to introduce Hawley, we'll begin with Hawley Helmbrecht. Hawley is a third-year PhD student in Elizabeth Nance's group, and she's also part of the Engineering Data Science Institute. She, along with Orion, is pursuing the data science option in chemical engineering. And we'll hear much more about her science and engineering shortly, but I wanted to note that she is president of the Association of Chemical Engineering Graduate Students. She's a senator in the Graduate and Professional Student Senate, and an executive board member of Women in Chemical Engineering, or WICI, which was founded at the University of Washington. Okay, and with that, I'll turn over the floor to you, Hawley.
All right. Hey, good afternoon, everyone. As I was just introduced, my name is Hawley. I'm a PhD student in Department of Chemical Engineering, and I have the great honor of doing my research in the lab of Elizabeth Nance. And over the past year, I've really been working on this project, which is the application of a robust framework for neuroimaging-based experimental routines for integrating domain and data science. It's something that I have named FIBER. And I wanted to talk about this more general project that I've been working on within the realm of a more specific project that we recently published in bioengineering and translational medicine. So that's how I'm going to begin. And in case you didn't guess from the title, neuroimaging, I study the brain. Oops, not going. I study the brain, and for the purposes of this talk, specifically the rat brain. And what I study within brain slices is what cells look like and what that means. And to narrow this talk even further, because there's a lot of knowledge about brain cells, I'm specifically going to talk about microglia, shown here in neon green, my favorite cell. And microglia are the resident immune cells of the brain, and they've become a pretty hot topic in science communication lately. So I picked a quote from Donna Jackson Nakazawa that describes how amazing they can be, that microglia, when triggered, can be destroyers, sparking a wide range of problems from memory loss and anxiety to depression and Alzheimer's. But under the right circumstances, they can be coaxed into angelic healers, able to repair the brain in ways that help alleviate symptoms and hold the promise to prevent disease. So when looking at microglia with an understanding of knowledge about what they do and what they mean, we can get a better insight into the greater brain environment. And so I specifically study their morphology. So why study the shape of microglia? Well, microglia have been shown to phenotypically change their shape in response to a variety of factors, such as those pointed out here by Dubellar in Frontiers in Immunology. Factors such as age, sex, species, and even what region of the brain these microglia reside. And more importantly, for this paper I'm going to get into talking about, microglia have been shown to change phenotype in regards to ischemia or oxygen deprivation in the brain, not only from the phenotype of a resident microglia, but also to an activated microglia, but also to the phenotype of macrophages alongside with monocytes inside the brain. And so this led to my hypothesis that oxygen glucose deprivation, a model of ischemia, induces detectable phenotypic changes in microglia that provide insight into the greater brain microenvironment. So how do we test this? Well, our methodology is through ex vivo slice treatment, which means that instead of taking an entire animal brain as one treatment group, we take the brain and we slice it coronally, and each slice is its own group. And those slices go through three different categories. They're either our non-treated sham control, or they are injured by going through half an hour of oxygen glucose deprivation, or they undergo treatment after injury. And all of that imaged on our confocal microscope produces beautiful images, such as those images shown here. And qualitatively, we can look at these images and see that there is a difference between our non-treated control, our treated, and our injured and our treated groups, such as the number of cells that exist on slightly their shape. But we're interested about studying this quantitatively, which is where I come in, and I analyze shape features such as their area, perimeter, circularity, major and minor axes of our cells, and their aspect ratios. And for this paper, we were able to have a really cool takeaway that after injury, our cells are significantly changing to a more circular shape, which often signifies an amoeboid microglia or a microglia that's dying. But with the treatment with azithromycin, that reverted back to something that's more similar to our non-treated control. Alongside these shape features, we also use vampire analysis, which is a visually aided morphophenotyping image recognition analysis from the Dennis Wirtz group at Johns Hopkins that allows us to take our cells and their outlines, feed that into principal components analysis, and creates for us this dendrogram of representative phenotypes. Basically, these 5 average shapes across our entire training group, and then gives us the frequency of how often those shapes show up in our testing group. And this also showed us pretty interesting information that the frequency of our representative phenotypes changes when injured, and that seems to revert after the treatment with injury. So that's quite exciting to show that treatment was helping and that there is a detectable phenotypic change. But as I said earlier in my talk, we're using microglia in order to get insight into the greater brain environment. And so when you look at these pictures, is only what I've shown you, you can only see the 2 stains that we're using, which is the neon green for microglia and this blue for an all nuclei DAPI stain. But microglia exist within a much greater environment. Here they're stained in red. They actually exist amongst other brain cells. And what I didn't really say is that we've also injected nanoparticles into all of these slices as well. And so what I've been doing is only a cog in the machine of a greater understanding of the brain environment. But to understand how that works, I have to introduce you a little bit more to the NANCE lab. So the NANCE lab works in 4 main areas. Number 1, we develop the slice culture models, such as the oxygen glucose deprivation model I talked about earlier. 2, we engineer nanotherapeutics so we can actually treat brain disease. 3, we measure the transport of those nanotherapeutics in the brain because we're chemies and we love transport. And 4, we characterize brain injury, what I really talked about. But to do such complex work in the brain takes a lot of collaboration. So this paper, and the results I've been talking about, actually took 9 independent researchers that have worked with or trained 17 undergrads and 7 high school students, and that use techniques, methods, materials from 9 collaborators across 3 institutions and 5 departments. So for my hypothesis, oxygen glucose deprivation inducing detectable phenotypic changes that went along with this methodology, it was actually only a smaller part of the project that I had what I already hinted at. We had nanoparticles also injected into all of these slices, and we're studying their interaction as well. And so we had a much larger hypothesis that oxygen glucose deprivation alters features of the brain microenvironment, which can then be leveraged by nanoparticles for investigation and treatment, shown through results from my lab mate Andrea Joseph, who studied flow cytometry to also show results that in injury, there's higher nanoparticle uptake that is reverted slightly with treatment. That goes along with my cell morphology that talks about circularity increasing with injury. But to effectively complete such interdisciplinary work that encompasses both data and domain science, we needed a robust method. So I created FIBER, or the Frameworks for Neuroimage-Based Experimental Routines. And the first step of FIBER was to develop data awareness. Everyone that was working on this hypothesis works with a ton of data. One experiment for us, sometimes one animal, can actually generate 1 terabyte of imaging data. But most of our experimentalists don't see themselves as data scientists. So we begin each of these explorations with a visualization of, so you are a data scientist now. And I actually take them through an exercise where if you resonate with the picture on the right, you click the red button, on the picture on the left, you click green. And we talk about questions such as, who do you think a data scientist is? And what environment do you think a data scientist works in? And although it may seem stereotypical, of the 22 people that I've done this with, almost all of them chose the picture on the left, even though even the data scientists in our lab work in an environment on the right. And then I think the most important question that we really delve into as a lab is, which types of data do most data scientists handle? And every single person that I've gone through this with chooses the numbers on the left, when it could really be anything. And so we discuss, what is data? It's not just our final numbers, but it's all of our images, it's all of our processes, it's the people that go into the process. And after we've discussed and developed a data awareness, we design data management plans. But in order to design an effective data management plan, we need a holistic process that is consistently updated and specifically implemented. So what I mean by consistently updated is that we don't create data management plans for individual experiments. We constantly update our data management plans. And I do this with data management workshops and creating tasks and worksheets and going through a constant iteration of familiarizing ourselves with relevant data science vocabulary. And then we create preliminary data management plans for every single consistent methodology in our lab, as well as regular data storage assessments of even preliminary data, even example data, even training data. Where is it going? How much is it? And in what files and forms are they in? And then quarterly, my lab mates are able to create these visuals, such as this by Mike, where he really said, these are all the variables we can control. This is what we work with. Or from an undergrad, Victoria, who helped us visualize our behavioral testing for animals. Or Andrea, whose data you saw earlier, who sees data management in a very different way and walked through the bigger steps. This allows us to have a better understanding of what our experiments mean data-wise. But we also want to apply this specifically. So we took all of those things we'd already created, all the standard methodologies we had from creating the size culture models and from working with nanoparticles regularly, and then applied them here specifically for this experiment. Once we have a data management plan, we're able to determine an optimal experimental pipeline. But for us, an optimal experimental pipeline for neuroimaging work with large amounts of image processing necessitates both domain and data science expertise. That we need the experimentalist expertise about how they captured their images and what that means in regards to their original methodology in order to inform data science feature addition. And we actually do all of this in Jupyter Notebook. We sit down for long meetings where we really work through every single step, and we write all of them out, and we say all the experimental steps that are necessary, all of the data science steps that will need to exist, who's going to do it, where are they going to keep their data, in what files, and for neuroimaging, what regions of the brain are we interested in, what confocal settings are we keeping up with, and how many images are going to exist for a specific analysis pipeline. And after we've done that, then we can finally build out a data science infrastructure. So I have a package that I'm currently working on, which is an if thresholds package that allows us to better threshold and segment cells in tissue, as well as integrating an altered vampire methodology from the Johns Hopkins group, because we also needed to apply that in tissue. And from there, we finally do our imaging. So we perform primary and supplemental imaging with our experimentalist expertise. But due to the FIBER process, we have multiple researchers, about 5 of them who are doing the imaging for this paper, produce hundreds of images that already have a data storage plan. They already have metadata conventions, so I don't have to go back in and delete 100 spaces inside titles. And they already know where they're going to be stored and how, which allows us to move quickly into producing interpretable visualizations of results, such as this one, where we interpret our dendrograms in a different way, that we can see that we actually get an increase in microglial heterogeneity with injury, which is then reverted with our treatment. And to get this one image actually took a back and forth between our data scientists and our experimentalists, 7 iterations before we decided that a bar graph where we actually hand put on every single dendrogram representative phenotype at each place was the best way to tell our story. But for us to get fascinating results using data science methods and neuroimaging processing, such as the results where we do see a change in the representative phenotypes with injury in regards to treatment, both our domain expertise and our data science expertise needed to be provided equitable voices during planning. That this methodology is a very experimental methodology, but doesn't exist without the downline data science pipeline. And Fiber allowed us to do systematic work and plan together. But the future of Fiber lies in both the empowerment of our data scientists and of our domain scientists and the freedom of our data scientists, meaning that we want to be able to apply this framework without needing to have our data scientists there. It's a pretty robust framework. It's neurological model agnostic, which is really exciting. So we're applying the same framework into an autism spectrum-like disorder, and in the future, hopefully in a traumatic brain injury as well. It's species agnostic, so we can do translational preclinical work a lot easier. So we're working in the rat with hopes to work in the ferret and pig pretty soon. And it's agnostic of cell type, which is really important when the brain has more than one cell. And it's also an iterative process. So after doing this paper, we realized that in order for our domain scientists to be able to use the pipeline, they needed to be trained, and they needed to be trained in a robust way. So we created what is called Textile for domain scientist education, and it stands for Tutorials for Experimentalist Interactive Learning. And we sat down and we really thought about what is the minimum amount of data science that our experimentalists need to learn so that they can use an image processing pipeline to analyze their own data. And I created this system of modules that are being moved to an asynchronous platform now and hosted completely through accessible Jupyter notebooks. So this is where Fiber has ended up now, is a 7-step process that allows our data scientists to explore new features and image analysis methods while supporting our domain scientists and effectively applying previously built data science infrastructure to future projects. Because I like to give insight into where we're going, and you saw Andrea's flow cytometry results earlier, our next step is to analyze the phenotype of microglia and cells that have actually uptaken nanoparticles. And so we're really excited to see about the differences in phenotype of cells that do uptake nanoparticles, as well as the phenotype changes once nanoparticles have been uptaken. And because I have such an amazing lab, I want to just thank the Nance Lab. It's an amazing community, as well as everyone in bold who took part in this paper, and all the undergrads and high school students that support all of the work that we do. And I believe now I take any questions.
Thank you very much. You know, I love to clap, but it never comes through virtually. So yes. That was really fantastic. So we have one question, and actually, let me bring it up. So the question is about when you talked about data management, that can mean a lot of things to a lot of people. You know, databases, various kinds of storage file formats. Can you elaborate a little bit more on the data management tools and how they play a role? And then I personally have a follow-up to that once you've kind of laid out some of the.
So my interest in data management started actually one day where I walked into lab. One of my undergrads went, "We're trying to download a 20 gigabyte file," and the computer said it has 1 gigabyte left. And we had entire experiments stored on that computer, and so I kind of freaked out. And so our data management really started quite elementary, based in just saying we need to keep track of what's happening. So that's where we started. And then next it started of being we have a very wide variety of data that I really didn't hit here, in that we not only have all of our images, but all of those images are correlated with brains that are stored in our freezers. And all of those brains are correlated with genetic data, which is stored in a different location and in a different file type. So at the beginning of a data management plan, it's getting researchers who haven't really had to think about data in this way just to physically, like with a pencil, map out where all the data is and what those data file types are in. After that, it moved into an economic analysis. So we had some of our data stored, a lot of our data stored in Amazon Web Services, because we were using some of that. So we started creating Excel sheets of saying, where does it make sense to use this data? How accessible does it need to be? Are we using cloud computing? So far it's been quite a lot of kind of grassroots, just sit on a whiteboard with people that haven't had to think about these things before and draw connections. Through EDSI, the Engineering Data Science Institute, we're working with a bunch of other people that are also having the same issue. And so we've been working, well, we were working with Magdalena Balazinska's lab on building up databases to actually keep track of our experiments as we go through. So I've been learning SQL in order to better help our lab mates track which brains are used for which experiments and how that tissue was then, so because there's a lot to it, but because we take a whole brain and then we slice the brain, one of those slices often goes to my analysis. One of those slices often goes to a genetic analysis. One of those slices can go to an RNA analysis. They can go many different ways, but they're attached to the same brain, or they can even go to different experiments, but we need to keep track of the original brain. So we've started the beginning to build out databases to keep track of, here's the brain it came from, here's the processes it went through, here's all the information that we have on it. And this is unlocking our ability to do retroactive analysis on experiments that we've already run that we haven't done the analysis of. This has nanoparticles and we have shape data. How did that shape of the cell change with regards to nanoparticles? So it started very grassroots, and now it's moving into a database management system using SQL. But the hardest part was that visualization of how much data do we actually have, and moving forward, what are we really going to do? We have a lot of people in a lot of different areas. So I hope that answered the question.
Well, I think it did. And then you also answered my follow-up question, which was about how do you stage data for retroactive analysis? So I think that was awesome. So there's another question that we have. Sorry?
Mm-hmm. Well, we've done more on that. So I like to name things after textiles. So I have fiber and textile, but we're also moving into something we're calling denim, which is to build up databases of experimental neuroimaging material. And so we've actually started training each of our lab members on how to keep track of their metadata, so that after experiments are done, they're actually uploading their images in a way that is organizable. I found a few papers from Nature, that synthetic bio, how they keep track of their data protocols, as well as their metadata, so that we can go back and say, this was used for this experiment at this time when we were using like this sort of work. And that's how we're really unlocking retroactive analysis, is by moving forward to just build in that habit of, once you're done with an experiment, upload it, and all the metadata that you think you'll never forget, but you forget 2 weeks later, please put that down in there. So it's a whole other aspect of what we're doing.
That's fantastic. So we have another question. So you talked about the educational components towards the end of the talk, and the question is asking if you can elaborate on that a little bit, and what, you know, the targeting for that is the audience, because you had undergrads, grads, postdoc, you know, you have a wide range of folks working on this. Is the educational components targeted any particular way, and how are you organizing those?
Yeah, so textile was like my passion project over the summer. And basically, I mean, you were all there, COVID happened. So I was supposed to take on 2 undergrads. And then COVID happened and our experimentalists couldn't take on more undergrads and high school students. And we didn't want them to not go anywhere. So I ended up taking on 7. And I all of a sudden had 7 mentees plus my normal 2 master students that I was mentoring, and I didn't know how to do it efficiently. And then we thought about what was the most useful thing to do. And so we started like thinking about, well, I need to train them in data science. None of them have any experience in coding, but all of them wanted to be in the lab for the therapeutic side. So I don't want to just teach them. I don't want to just say, here, learn Python. And so I sat down with a few of our experimentalist grad students, and I was like, here's the pipelines that you would most want someone to run for you later. This is the training I need to go into it, which was the training of what is data science? Are you a data scientist? And the basics of our image processing pipeline, which is like kind of an introduction to scikit-image. I actually talked to scikit-image a little bit at the Scientific Python conference, and I've been meaning to make a tutorial for them based off some stuff I'd done in textile. But so it's very easy to understand. And I've done that because I split it into 3 concepts, which is the first concept, which is what you explore by yourself, which is a very philosophical exploration of what is data science and what is an image. Because most people don't even know that an image is a matrix of numbers, which is okay to not know that if you never had to deal with it. And then we do a 1-hour component together where we walk through this small part where they code it themselves, and I help them go through issues. And it's all in Jupyter Notebook because we found that that's like people aren't as afraid of that if they don't know any coding. And then we do a recall activity where I have them apply it on an image they already have, or like a new image. And we've been doing a feedback process where everyone completes a poll and then reiterates that. And I originally had it targeted only for high school students, but then the PhD students in my lab really wanted to do it. So then PhD students joined on. And then when we were working with collaborators, a few collaborators were like, well, I would like to learn just enough so that I can understand what's doing in image processing. So we've actually had like high school students to PhD students plus some do a sit-in on 1 or 2 that have found this to be quite an accessible way to just learn what they need to know for their experiments to be a little bit better, but not have to spend like 3 months really in-depth learning how to do Python.
That's fantastic. I'm blown away, and it strikes me, you know, Nicoletta is also running the seminar with me, and you know, Nicoletta deals with image data. It might be satellite image data, but it's, you know, the concepts that you're trying to adapt and share with folks are probably very transdisciplinary in a lot of ways.
I just wanted to say that they are very inspirational too, and quarterly meetings for data management sounds like a really great idea.
I know.
I have a colleague who says that metadata is love letters to the future, so I think that's really
it's so much fun. When I had my lab members first draw up what their like metadata looked like and what their process looked like, I had a way that I expected them all to do it, which is the way I did it. And when they came back with just this spectrum of viewing data in a different way, it was so amazing. Like one of our undergrads also included things like safety protocol data, and someone else decided they wanted to include chemical, like the numbers on our chemical bottles, because they'd at one point had an issue with contamination in a previous lab they'd worked in. I was like, these are like amazing ideas I never would have run into myself, except for the fact that we like all did this separately and brought them together. And so it was like a really fun experience for me to see how they viewed data.
That's fantastic. So we need to move on to our next speaker, but Hawley, there's more questions for you. Can I ask you to stick around until the end, and then we can continue with some of the questions for both you and Orion?
Happy to stay.
Awesome. So I'm going to give Orion a